for the invitation. So sorry about that on the sudden. Yeah. It's really nice to be here to attend this meeting. I'm also very amazed about the quality of the talk presented in this meeting. Today, I'm going to share with you some of the work we have done over the past 10 years in Asian population to identify genetic factors for breast cancer and colon cancer. I thought I choose this topic because I think it will be helpful to kind of contribute to the discussion about how human diversity contribute to the identified novel genetic factors for complex disease. So before I start this talk, I'd like to present this slide, some background information. We know genetic factors are important for breast cancer and colon cancer risk. So family history of breast cancer or colon cancer is one of the most consistent identified risk factor for this cancer. The risk all depend on the family history pattern and the number of people in the family diagnosed with cancer and age at onset. And about 10 to 15 or 20 genes have been identified with this pathogenic mutation really confirm a very high risk of cancer. Risk can be very high, kind of like breast cancer, lifetime risk can be about 80 or 90%. But pathogenic mutation in a general population is very uncommon, it's very rare. So these mutations explain a very small percent of the cases in the general population. So it has been hypothesized that most of the heritability of cancer and some complex disease can be explained by those low pathogens but common genetic variants. So I think started in about early 90s, about 30 years ago, many studies used this carrier gene approach, look at a couple genes, a couple of variants at a time, look at those variants related to cancer risk. And many studies have been done during about 15 years period. And most of the study report some positive association, sometimes report no association. Overall, these results really cannot replicate it. So recently, actually not recently, we summarized the status of these carrier gene studies and show that just large number of publications and very few of these variants can be replicated. So since 2005, this Genome-wide Association approach emerged as a very promising approach to identify genetic factors for complex disease. Some of the cancer may be caused by gene-gene or gene-environment interaction. So today, I'm primarily talking about GWAS, Genome-wide Association Study. So we started a pilot study of GWAS in 2005. We used these beta testing chip is before released to the public, and then they asked Vanderbilt to test this chip for them. So now we think about GWAS is simple, straightforward approach. But at that time, about 15 years ago, this is still very new. There was a lot of doubt in the community about whether we really can use the GWAS approach to identify genetic factors. So that's why we did this pilot study. We want to look at study feasibility and then set up the pipelines for analyze data. All of a sudden, we need to analyze 500,000 variants at a time, not like in the past, we just look at a couple variants. And also, we need to show some of the proof of principle data, so really we can find those signals in this haystack. So we did 200 case system controls. Actually, it's quite expensive at that time, about $1,300 per sample. So I spent about $260,000, which is actually one of the most expensive pilot study ever done. And so we found some evidence. This is the different p-value cut, and this is the number of variants. They are significant in this threshold, and then this is just expected under null hypothesis. And you see that this number observed always bigger than number expected, particularly in those regions with strong evidence for harbor breast cancer gene. This is breast cancer with breast cancer genes. So with this data, so we submitted our one grant in early 2006 actually, not 2016, 2006. So before our grant was funded, there were 3 GWAS papers published in the spring of 2017. One from Cambridge, led by Dr. Easton. This is called the Breast Cancer Association Consortium. BCAC is the largest international consortium for breast cancer research. Another one led by Stephen Chanick and David Hunter for NCI and Harvard combined program. And the third one, the Decode in Iceland, is actually a company. So they publish all these studies. So for example, this one Cambridge study, they did a 3-stage. The first stage, they analyzed about 26,000 variants, which this number certainly, something will be statistically significant just by chance, right? So they need to do replication. Stage 2 analyze about 13,000 variants, and stage 3 analyze about 30 variants, and in the end, they find the 5 SNP that reach genome-wide statistical significance. And same for other 2 studies, they find something, and you can see that the results actually kind of can be replicated. They find kind of similar association. So our proposal, that time still in study section, and some reviews asked when is still needed, to do a study in Asian population. There are 3 big GWAS in European descendant. Why we need to do a study in Asian population? So we argue that study in Asian population may help identify additional novel genetic variants for breast cancer. And then particularly some of these variants that are difficult to identify in European descendants, because just the genetic architecture difference between population and also background exposure may be different. So that's one. And then we can evaluate the consistency of the association and evaluate generalizability of the findings from European descendant to Asian population, and identify some unique genetic variants for Asian population. We can fine map the loci, identify possible causal genes, because using the genetic architecture difference, so we can narrow down the LD block. And also we can identify some more specific variants for Asian population. With this consortium, we can promote collaboration, bring people with different background, expertise, and we can promote interaction. Certainly with the larger sample size, we can have more fundings. So another thing we argue is that the study in low-risk population may have higher statistical power to study genetic association. In the interest of time, I may not go here, but if you're interested, I can explain that to you later. So the study was funded in 2008, and then we currently we include about 80,000 cases and controls from about 20 studies, most of them from East Asian populations. We certainly like to include population Indian, but so far we have not identified collaborators. Certainly we'd like to do that if people are interested to be involved in this consortium. So we identify about 70 loci with these P5 times 10 minus 8, which is the GWAS significant level, including about 40 new loci. And this consortium become the second start in terms of productivity is number 2, and the other one is the BCAC I mentioned earlier. So here just some of the publications, including one actually just get accepted in Nature Communication recently. So in the interest of time, I'm going to provide 2 examples to present here to show that actually these loci are very important that identify in Asian population. So this is the first study, first paper we published. We have a discovery stage and find that heterozygous, homozygous, there's a significant increased risk. And we did 2 replication, quite consistent funding, and then the pool analysis is about 10 to the minus 15. So it's very, very small p-value, quite definitive funding. So this is located in 6q25 near a gene called ESR1, and then people know here it's coding the estrogen receptor alpha, which is very important for breast cancer. So we did analysis stratified by ER status. This is ER positive, ER negative, and found the association actually stronger among for cases with ER negative breast cancer. So this is the first genetic locus identified for ER negative breast cancer. As mentioned previously, ER negative breast cancer actually have a poor prognosis. So then with those findings, we want to look at the association, whether we can replicate this association in European descendant or in African descendant. And we found that in other Asian populations, including Japan, Korean, we found similar association as we found previously in Chinese population. In European descendant, there's a weak association, and in African American, actually there's no association. So which suggests that the variant we identify is not a causal variant, just a tagging SNP, the ability to tag the causal variants differ by population, right? So we did some in vitro experiments and identified 3 SNPs that located between this open reading frame with the ESR1 genes, and we did some analysis and found this SNP may show some functional significance. And we go back to the population and find this SNP actually show consistent association in all 3 populations. So this locus is very important because this is in the ESR1 gene area that attract a lot of attention by other group. After our study was published, and many other studies was also look at this locus. This is just some example. One study showed that there are 2 independent association signals in this region, and another study showed that the 2 open reading frames may interact with estrogen receptor alpha, and that may explain some of the resistance to hormone therapy for some patients. And mammography density, this locus is actually also related to this density, which is a major risk factor for breast cancer. More recently, the BCAT study showed that at least 5 independent signals in this region. And each of these signals related to one type of breast cancer defined by ER, PR, and HER2 status. So pretty much all the study, GWAS, conducted look at single nucleotide variants because this is relatively easy to do. But structure variants can be very important because they explain more difference between 2 individuals in terms of their sequence. They affect a large fragment of the genome, more likely to have functional importance. And they likely explain some of the missing heritability from the GWAS of the single nucleotide variants. So we conducted the first GWAS of structure variants. We focused on copy number variation, which is a major form of structure variants. We use FA matrix 6.0 array, which contain about 1.8 million variants and probes. We scan the whole genome and identify a promising association for replication. So in stage 2, we use qPCR to confirm the association. And total is about, I think it's about 13,000 cases in controls. We identify a deletion, a common deletion in ApoBac3, this region, that confirm very high risk of breast cancer. And people work on GWAS, this odds ratio 1.77 is really high. So far, this is still the strongest association identified for breast cancer. So this deletion is quite interesting. In East Asian population, the frequency is about 35 to 40%. The deletion frequency is about 35 to 40%. In European descendant, it's about 10%. In African, it's about less than 1%. So that's explain why GWAS in Asian population actually identify these variants because we just have a lot of statistical power compared to study conducted in European descendant. But after we identify these, we look at just in European descendant in this particular locus, and we found quite consistent association, quite high odds ratio. So recently, the BCAC International Consortium replicate this finding using tagging SNP. Actually, they didn't look at the copy number. directory and analyze the copy number directory. They're just using the tagging SNP. So that kind of explains why the association is quite weak in the European descendant in that report. So it's turned out this ApoBac3 is very important in human cancers. This is a report from Michael Stratton from the Sanger Institute. They published a paper in Nature in 2013. Look at mutation signatures in many cancers. So what they find is one signature caused by overexpression of ApoBac3. I think people cannot see that. Is here, maybe signature 2, and also I think another signature, maybe signature 14, some here. Actually, you can find these signatures in many cancers. It is actually the most common mutation signatures ever identified in human cancers. So we were kind of concerned because what they found is the overexpression of ApoBac3 related to this mutation signature. And what we found previously is a deletion. So what happened? Seems the story is not consistent. So we went back to look at our data and then confirm that our data is correct. It's turned out that this deletion cause a fusion gene and then bring the 5' and the coding region together and create this more stable these mRNA, and then that causes a lot of replication and translation. So that kind of explains overexpression of this gene in tumor tissues. So this is an interesting story from population to kind of fundamental biology. So let's talk about this colorectal cancer consortium. I only have a few slides about this. This started in 2009, and so far we have about 70,000 cases and controls, primarily also from Eastern Asian population. We identify about 40 loci. Actually, this is more than this. 35 novel loci in this consortium. So before this paper was published, actually, our study contributed more than 1/3 of all the loci identified for colorectal cancer, just European descendant, African American, all put together. So we account for more than 1/3. There's some reason for that, kind of methodology, population uniqueness, but in the interest of time, I'm not going to talk about this. So these findings uncover some of the novel biological pathways for colorectal cancer, which I listed here. Some of these actually point to those pathways we know already, like Wnt signaling pathway, but some of these just novel pathway like fatty acid metabolism or sex hormone region. So this really GWAS can uncover novel biology for human cancer. So far, this GWAS has been very successful, identified about 52,000 unique these trait variants association. For cancer, I'm sure it's over 2,000. So what we're going to do with this data? What we have learned from GWAS, one is the multiple common risk variants exist for virtually all cancers. Whatever cancer you look at, there are multiple variants. With a few exceptions, example AQ24, which is near the MYC gene variant. Those variants identified is more cancer specific and sometimes kind of cell type specific. And most variants carry very small risk, like odds ratio, usually a leading odds ratio less than 1.15. And then they explain so far a small fraction of the cancer heritability. They reported primarily in the uncoding region, regulate gene expression. We need to do study multiple populations to really uncover the genetic background for complex trait. Studying multiple populations to really uncover the genetic background for complex trait, show promise in identify high-risk people for cancer prevention. So what next? What we're going to do next? So one is that we need to expand genetic studies in non-European descendants, and most of the studies so far are conducted in European descendants, and we need to do that in other populations. So this is a study we started about 3 years ago in African American breast cancer. The reason to do that, as mentioned yesterday, African American with breast cancer, they have high mortality rate, young age at onset, and then they have a high rate of triple negative breast cancer. And African genomes are very diverse, so they can be very informative if we study particularly rare variants. So we pay a large grant for NCI to launch this study, including about 20,000 cases, 20,000 controls. So we used that's the first honor studies in African Americans conducted in the US, and then we also include some study in Africa. We also try to understand possible mechanisms of the association between germline variants and then breast cancer risk. So this is other ongoing work. We try to search for real variants or structural variants using next generation sequencing, work to characterize GWAS identified loci, using GWAS identified association variants as instrument in epidemiology and clinical study. As many of you may be familiar with called Mendelian randomization analysis. We're also doing some integration of multi-omics data to understand GWAS finding and identify novel disease biomarkers, and try to evaluate gene-gene interaction, and to continue to improve risk prediction using polygenic score. So I just want to show you one example how we combine genomic data with other omics data to identify novel genes, novel biomarkers for cancer risk. So this one is we call Transcriptome-wide Association Study. Here I call GWAS. The question is, what's the associated gene expression in breast tissue related to breast cancer risk? So we know it's very hard to do study, and the easiest way to do is that we identify cases, identify controls, and then we look at the gene expression in the breast tissue and then compare the difference. But it's hard to do. What is normal tissues you're going to look at, right? Normally we get adjacent normal tissues, but that are not normal. And then controls, what are the controls? Normal healthy woman may not necessarily willing to give you tissues for your study. So it's hard to identify cases, controls, and also it's kind of expensive. You profile the whole transcriptome can be very expensive. So fortunately, some of the gene expression can be predicted using genetic variants. So this is actually what we did. We use data publicly available from GTAX and TGA to build models, genetic models, to predict gene expression. And then we used these models, apply data to the GWAS data from Cambridge. This is BCAC data from Cambridge. It's about 230,000 cases controls. And we just look at this association. We identify many significant association and kind of gene expression with breast cancer risk. And also we select a few genes, about 14, 15 genes, and do some quick analysis, and show that many of the genes actually have some cell function, affect the cell function.
Professor Zhang, you have to wind up.
Okay, yeah, I'm almost done, yeah. So we also completed the studies for other cancers as well. So this is summary. Genetic association study in Asian population provide a unique opportunity to identify novel susceptibility factors for cancer and other complex traits. I hope I convince you about that. A large number of novel risk have been identified for breast cancer and colon cancer using GWAS conducted in Asian population. This is complementary to the European descendant study. And using this omics data, so I just show you the transcriptome data, GWAS, and we actually have done study about methylation, about protein, metabolomics, all show a very interesting association, identified more than 500,000 novel association. So I don't have time to present here. Research consortium put all data together, offer a very powerful approach for scientific discovery. Thank you very much for your attention. I'm happy to